Welcome to biology. This is Mr. Troll. Today we're going to be taking a look at sex-linked traits and pedigrees as part of your genetics unit study. So the first thing we want to answer is what determines sex? So you guys are familiar with karyotypes. You have learned about karyotypes. We did a lesson on karyotypes. You guys had a karyotype lab. And so as far as sex is concerned, if we're looking at a human karyotype, where we want to focus is on this 23rd pair of chromosomes right here. So if you recall, we looked at the 21st pair to determine things like trisomy 21, which is Down syndrome. So if there's 3 chromosomes there, an individual has that genetic disorder. But for sex, we want to focus on the 23rd pair. If there are 2 X's, then that indicates that the sex of the individual is a female. If there's an X and a Y, then that tells us that it's a male. And so this karyotype, since it has XY for its chromosome pair number 23, that tells us that this is a male. We could further describe this karyotype by looking at the 21st pair and saying that this is a normal male. So that's what our focus is going to be as far as sex-linked traits, is we're looking at traits that are inherited on that 23rd pair of chromosomes. So some traits are just found on the X. So one of the tricks that I like to use to help you guys remember this is when you see sex-linked, I want you to think of that as, and really focus on that X as X-linked. So sex-linked traits are located on the X chromosome. So we just established that if it is a male, they have an X and a Y, and that a female has an X and an X. So these traits are only found on those X chromosomes in the 23rd pair. Now, this one important thing to understand is that this really doesn't have anything to do with sex. So all chromosomes are inherited via sexual reproduction. The reason why these are called sex chromosomes is because they are the sex determiner. There are 2 traits that we're going to be examining that are inherited on this 23rd pair on the X chromosome of the 23rd pair, and that is color vision and also blood clotting. So we'll look at color blindness first. If someone has color blindness, they have the inability to distinguish between red and green when they are close together. So if you take a look at the diagram on the right-hand side, if you were to go into a doctor's office and they were to inquire as to whether or not you were colorblind, then they would give you a test similar to the one seen on these diagrams. And so if you're not colorblind, then you can see these numbers here, okay? 25 there, 45 there. We can see a 6 right there. We can see an 8 right there, a 56 on this one, and a 29 here. Individuals who are colorblind would not be able to see those numbers, and they would not be able to distinguish that because the background would look like just one big blur. Colorblindness is sex-linked, and it's also recessive. And so what we're going to do for our alleles is we're going to represent normal vision as a big B and colorblindness as a lowercase b. So for males, we have to include the chromosomes, and we're going to put XY for the males. So you can see we have XY here, XY here. Now, the reason why we have to include the chromosomes with the alleles is because this is the only pair of chromosomes where males and females are different. So if we look at any other pair, pair 1, pair 2, pair 3, pair 17, any other pair besides 23, males and females both have two X's. So up until now, we have not included the chromosomes in parts of our genetic problems because they have been the same. So we have just included the alleles. But that changes with the 23rd pair. Since males are XY and females are XX, we have to include the chromosomes as well as the alleles. So remember when I said sex linked, I want you to think of X linked. These alleles can only be linked to the X chromosomes. So males having an X and a Y for their 23rd pair can only have one allele. And that allele is represented as a superscript on the X chromosome. So since normal vision is big B and color blindness is little b because it's recessive, if we have a normal male, meaning they have normal vision and they're not colorblind, then we would put X uppercase B, Y. And if we had a colorblind male, we would represent that by X lowercase b, Y. So males only have one allele for sex-linked traits. And this is only for sex-linked traits. I will caution you, for whatever reason, I've had students in the past that after I show them how to work out a sex-linked genetics problem, they try to work out all problems this way, and you can't do that. They expect you to know that color blindness and another disorder we're going to look at later, hemophilia, are sex-linked. But if it's not color blindness and it's not hemophilia, and the question does not tell you that it's sex-linked, then you don't work out the genetics problem the way that we're going to work them out today. For females, we include both X's. Again, we're looking at the 23rd pair. And so females, since they have two X's and sex-linked traits are X-linked, females have two alleles for sex-linked traits. So if a female is normal, she has normal vision, she would either be X big B, X big B, or she would be X big B, X little b. So females that are normal for sex-linked traits can either be homozygous or they can be heterozygous. And if they're heterozygous, we use another word that's synonymous with heterozygotes, and we would say that this individual on the right here is a carrier. So that's another name for a heterozygous individual when we're talking about genetic disorders and sex-linked traits. Females can be carriers for these traits, however, males cannot. And then if a female is colorblind, which is extremely rare because she has to have two recessive alleles on her X's, that's the only way that she can actually exhibit that trait. With that being said, males are much more susceptible to sex-linked traits than females, especially recessive ones, because if males have the recessive allele, then there's nothing on the Y chromosome to be able to mask that. Where with females, if they have a recessive allele, there's a chance that they have the dominant on the other X, so that the dominant normal trait is what's seen. The other sex-linked disorder that we're going to learn about is hemophilia. And hemophilia, individuals with this disorder have a reduced ability to clot their blood. They don't have a clotting factor. And so we think of blood clots as being a bad thing, and they can be. They potentially are, depending on where those blood clots occur. However, when you get a bloody nose, as shown in the diagram, or a nick, a cut, a scrape, etc., or even a bruise where you have internal bleeding, that's very dangerous to people with hemophilia because they do not have the ability to clot their own blood. And that's essentially what makes an area stop bleeding is that those clotting factors basically plug the hole. We'll use the same type of letter assignment or allele assignment for hemophilia as we did for color blindness. Since hemophilia is also recessive, we will use a big H for normal, individuals that have normal clotting factors and do not have an issue with getting a cut or a scrape or a bruise. And we'll use the lowercase h to represent hemophilia. So same thing applies. This should look similar to what we just learned about a second ago. Males, we're going to have an XY. Females, we're going to have both X's because, again, this is a sex-linked trait. And we are looking at the 23rd pair, which differs. Once again, sex-linked means X-linked. So we don't ever put an allele on the Y. We leave the Y's alone. We only focus on those X's. So if it's a normal male, they would have X, big HY, since normal is dominant to hemophilia. And if we had a hemophiliac male, we would have X, little h as a superscript Y. Females, same rules apply as it did with color blindness. Females that have normal clotting factors can either be homozygous normal, as shown here on the left with X, big H, or they can be X, big H, X, little h. And again, we would describe that female as a carrier. Females who are hemophiliacs, which is much rarer than males who are hemophiliacs, have to have both little h's on both of the X's of the 23rd pair to be affected by this disorder. So again, this just reiterates some of the things that we have touched on, but males are more likely to be colorblind and males are more likely to have hemophilia than females. The reason why? Males only have that one X. So they only have one X on that 23rd pair. So they only get one copy of that trait. So again, if they get the recessive allele, there is nothing on that Y chromosome that can mask or hide that recessive allele. Where if females have the recessive allele, there is a chance that they have the dominant allele on the other X that can mask that genetic disorder. So they get two copies of the trait because they have two X's on their 23rd pair. So example number 1, we're going to work this out together, and I'm also going to show you the answer in a little bit cleaner fashion here in a second. But example number 1, we're saying that big B is normal and little b is colorblind. And then it says a wife who is heterozygous, and that means she has different alleles. So there's our heterozygous female. We know it's a female because there's our two X's, and she's heterozygous, so big B, little b. She is heterozygous for vision. What is the chance that they will have a child that is colorblind? So to answer this, we have to work out this Punnett square. So the same rules apply as with other Punnett squares. Whatever's on the top drops down into the bottom boxes. Whatever's on the left moves over to the right. And we keep X's before Y's if it's a male. And a good rule of thumb is when you run into a situation where you have a box that has both a dominant and a recessive allele, that you put the X with the dominant allele before the X with the recessive allele. So I'm going to bring this X big B right down here. I'm going to bring this X big B right over here. I'm going to do the same. So whatever's on the left continues to move to the right. Whatever's on the top continues to drop down to the bottom. And my Punnett square is complete. So I'm almost ready to answer the question of what is the chance that they will have a child that is colorblind. So to do this, what I want to calculate is my genotypic-phenotypic ratio. And so I'm going to look to see how many different genotypes I have right here. So I have one of these, and I have one of these. That's different. I have one of these and one of these. So I actually have 4 different genotypes. So I'm going to have a 1 to 1 to 1 to 1 ratio. So I'm going to have 1 X big B, X big B for every 1 X big B, X little b for every 1 X big B Y for every 1 X little b Y. And so now I'm going to look at what that translates into. This individual I would describe as a normal female. This individual would also be a normal female, even though she is a carrier. We have indicated that in the genotype, but we do not include that in the description of the phenotype. So I would say that I'm going to have or predict to have 2 normal females. This individual would be a normal male for every 1 normal male. And then last but not least, this would be my 1 colorblind male. So the question is, what is the chance that they will have a colorblind child? That is the only chance, that 1 out of 4. So the chance that they would have a colorblind child would be 25%. And that gives you the answer right there. Example number 2. A man has hemophilia and his wife is heterozygous. What is the chance this couple will have a son? So now we've taken it one level deeper. We're not asking about a child. We're asking specifically about a son. And so a man has hemophilia. So I'm first going to do my test cross. I'm going to make my man right there. X little h, Y. XY is the male. Little h indicates that he has hemophilia. And then it says his wife is heterozygous. So she is going to be X big H, X little h. So I'm going to separate those chromosomes with those alleles over their respective boxes. Whatever is on the top, once again, drops down. Whatever's on the left moves over, keeping the dominant alleles in front of the recessive when that situation arises, and keeping the X's in front of the Y's so that we're consistent. So we're looking at the chances that they will have a son with hemophilia. And so again, they here are the daughters. So we're only focused on the males, which are in these 2 boxes. And that is my only chance of having a son with hemophilia, is the bottom right box of this punnet. Now, the percentage is a little different. So notice that we didn't make that percentage 50%. We made that or 25%. We made that percentage 50% because it's asking if we have a son. So again, we're only focusing on the males. What is the chance that they will have hemophilia? And 1 out of 2 would be 50%. So that's how you would calculate that type of problem. So a little bit, just one additional step from the other problem that we worked out. So switching gears, last thing you've got to learn about today is a pedigree. What a pedigree is a chart of the genetic history of a family over several generations. And we use that chart to predict, offspring, or genetic counselors use this type of information so that a couple that's interested in having children can come in and they can determine if they have some of the same recessive traits, if they're carriers. So they're able to, so these scientists or genetic counselors are able to find out about your history. They're able to make a chart, analyze this, and give you kind of a prediction or probability of the genetic disorders that your child may or may not have a chance of acquiring from you and your significant other. Pedigree symbols. You don't need to know all of these, but there are some that I really want you to focus on and understand. And so the most important thing is to know how to distinguish a male from a female. Males are represented by squares. So anytime you see a square on this chart that I'm getting ready to show you, that represents a male. Females are circles. And so again, being able to distinguish what sex you're looking at is really important. If they are not shaded in, that means they are unaffected. So whatever genetic disorder we're looking at, if they're not shaded in, it means they do not have that disorder. It doesn't necessarily mean that they're not a carrier, but it means that they're unaffected by it. If they're shaded in, that means they are affected. So, for example, if we were looking at a pedigree that dealt with hemophilia or color blindness that you just learned about, and we had a square that was not filled in, we would say that we had a normal male or a normal female. Or if it was shaded in, we would say that we have a colorblind male or a colorblind female or a hemophiliac male or a hemophiliac female, depending on the trait that we're looking at. Okay. Whenever you see a line between a male and a female, that means marriage or mating. And so from that, you will see a bracket that comes down that will represent the children produced by that marriage or mating. These down through here, useful information, but we're not really going to focus on those. We're not really going to look at deceased either, so you can kind of ignore that. Sometimes you might see this, okay? So you might see half of a square or half of a circle shaded in, which tells you that the individual is heterozygous. However, sometimes when they're not shaded in, if it's a recessive disorder, they can be heterozygous, but it just doesn't indicate it by shading half of the square or circle in. So we're going to take a look at one of these. You guys are going to have some practice problems to do on these as well. So we have squares, we have circles. Some of them are shaded, some of them are not. And so the first thing we're going to decide is that this is a recessive genetic disorder. And the reason why I know that is because we can look at these parents, parent 8 and 9. So 8 would be mom because it's a circle. 9 would be dad because it's a square. And 11 would be their son because it's a square. So that bracket joining them indicates mating. And then this bracket coming down indicates that that's their child. Over here on the left, we have generation 1, 2, and 3. So this pedigree spans 3 generations. And then the numbers, the individuals are numbered accordingly. So we would, if we were asked to identify this individual, for example, it might refer to that individual as 1, 1. Or this individual, for example, it might refer to that individual as 2, 4, that sort of thing. Okay. So the reason why I know this is recessive is because the mother, number 8, is unaffected, and the father, number 9, is also unaffected, but their child is affected. And so the fact that 8 and 9 hid this disorder tells me that it's recessive. The other thing that I'm going to look at and compare is the number of males compared to the number of females who are affected by this disorder. So if I look at individual 1, affected, 3 is affected, 6 is affected, 10 is affected, 11 is affected. So there is 4 males that are affected and only 1 female. That is a disproportional number, which tells me this is sex linked. It tells me this is sex linked. So one of the questions that you might encounter for this is you might have to determine the genotypes of any of these 11 individuals. So now that we know that this is a sex-linked trait, I'm going to go ahead and put XX for all of my females, and I'm going to put XY for all of my males, because since it's sex linked, I have to include not only the alleles, but I also have to include the chromosomes, because the 23rd pair is different for males and females. The other thing I'm going to do is I'm just going to use a very generic allele assignment. Since we've said that it's recessive, I'm going to make big N normal, and I'm going to make little n affected. And that could be color blindness, since it's sex-linked, or that could be hemophilia. It doesn't really matter. For our purposes, we just want to calculate the genotype. So for individual number 1, that's an affected male. So that would be X little n Y. Individual 2 is unaffected, so I know that she is going to have at least 1 big N. And I can pretty much do that for all of my unaffected males and females, is give them all 1 big N. And the reason why I can do that is because if they're unaffected, they have at least 1 big N. Now, for my males, once I give them a big N because they're unaffected, like I did with individual 5 and individual 9, then I'm done with that. I don't have to do anything with that. The other thing I can do is I can go ahead and look at my males who are affected, like individual 1. Individual 3 is like that. I can give individual 3 a little n, and I can give individual 11 a little n, because number 11 is affected as well. Number 10 is one of those rare unique cases where I have an affected female. So the only way she can be affected is by having 2 lowercase n's. Now, I've already established that 11 was affected, but the parents were not, which means that trait was hidden. And so since the male is normal and the Y can't carry anything, then that little n received from number 11 had to come from the mom. So mom would be heterozygous, or mom would be a carrier. The same thing applies to individual number 7. Individual number 7 is normal, but the female is affected, which means that female had to be able to give one of those little n's. So she would also be X big N, X little n. She would be a carrier or a heterozygous individual, just like number 8. Number 6, missed that guy earlier. That's the same as individual 1, 3, and 11. We would say X little n, Y. 5 is done. And then 4, we have X big N. She got one of those X's from mom, and X little n, because she had to get one of those X's from dad, and the only one he had to give was the X little n. Individual 2, if we look at this and we see, we look at their children together, and we see that number 6, the male, was affected. Well, number 6 got their Y from dad, which means that X little n had to come from mom. So most of the pedigrees that you encounter will be dealing with sex-linked recessive traits, and what you have to determine is the genotype of those individuals.